Assalamualaikum and hi everyone. We are from Group 4. We present Immune System. This is our group member Atika, Hanisa, Himalata and Anas Adel.
Immune system is one of the most important systems in our body to ensure that the body can function properly and stay healthy. Immune system is a complex network of cells and proteins that protects the body from possibly harmful substances by recognizing and responding to antigens. Antigens are substances on the surface of cells, viruses, fungi, and bacteria that are usually proteins. Antigens also include non-living substances such as poisons, chemicals, medications, and foreign particles, such as a splinter. The immune system is capable to keep the record of every germ it has ever destroyed, so that it may rapidly recognize and eliminate the microbe if it re-enters the body. Abnormalities of the immune system can lead to allergic diseases, immunodeficiencies, and autoimmune disorders. Anatomy structure of the immune system. Skin and mucous membranes are the first line of defense against germs entering from outside the body. Antibacterial substance can kill germs right from the start. A certain enzyme found in saliva that aerates and tear fluid destroys the cell walls of bacteria. Mucus, the bronchi helps trap many of the germs we breathe in, so they can be moved out of the areas by hair-like structures called cilia. Stomach acid stops most of the germs that enter the body in the food we eat. Harmless bacteria on our skin and many of the mucous membranes in our body also act as part of the immune system.
Lymphoid organs have two, which is primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs include bone marrow and thymus, which is bone marrow is a sponge-like tissue found inside the bones. That is where most immune system cells are produced and then also multiply. These cells move to other organs and tissue through the blood. The thymus is located behind the breastbone above the heart. This gland-like organ reaches full maturity only in children and is then slowly transformed to fatty tissue. Special type of immune system cell called T cell mature in the thymus. For the secondary lymphoid organs, it include lymph nodes, spleen, tonsil, and various mucous membranes. Lymph nodes is a small bean-shaped tissue discovered along the lymphatic vessel. They act as a filters. Various immune system cells traps germs in the lymph nodes and activate the creation of special antibodies in the blood. Spleen is located in the left upper abdomen beneath the diaphragm and has a different function, which is it stores various immune system cells. When needed, they move through the blood to other organs. Phagocytes in the spleen act as a filter for germs that get into the bloodstream. It also breaks down red blood cells and it stores and breaks down platelets, which are responsible for the clotting of blood. For the tonsil, they are located at the throat and palate so they can stop germs entering the body through mouth or nose. Tonsil contain a lot of white blood cells to kill germs. Mucous membranes. The bowel plays a central role in defending the body against germs. More than half of all the body cells that produce antibodies are found in the bowel wall, especially in the last part of the small bowel and in the appendix. These cells detect foreign substance, mark and destroy them. They save information about the substance so that they can react faster the following time.
Now I'm going to explain about the function of immune system in the body. There are three main functions of immune system which are to prevent the entry of pathogen, to fight diseases causing changes in the body, to neutralize and recognize the harmful substances from the environment. How do our immune system functions in our body? There are two categories of immunity, which is innate immunity and adaptive immunity. Innate immunity is a general defense against the harmful pathogens and performing the first line and second line defense, while the adaptive immunity is more focused defense against the pathogen and it will perform third line defense against the pathogen. Innate immunity is a non-specific response where it will happen after some minutes of pathogen's entry. The innate immunity will be carried out by phagocytes, natural killer cells, inflammatory response, complements, fever. The first line innate defense is known as surface barriers, and also the second line of innate defense is known as the internal defenses. While the adaptive immunity is the specific defense response, which will delay as 3 days for the primary immune response, but immediately respond for the secondary immune response. It will having memory which will lead to the faster secondary immune response. There are two types of adaptive immunity as well. First is humoral immunity by the B cells and then the cellular immunity by the T cells. Humoral immunity is an antibody-mediated immunity and defense done by the antibody that produced by lymphocytes. Cellular immunity is a cell-mediated antibody and defense done by the lymphocytes as well. Adaptive immune system will undergo the third line defense. It is a specific response to eliminate the pathogenic cells in our body. It involves the production of antibodies that produced by the lymphocytes. This response is also having the memories. These are the pictures that shows the adaptive immune system. Here it is the innate difference between the innate and also the adaptive immunity. While here it is the difference between humoral immunity and also cellular immunity. For humoral immunity, we will have the B cells, while the cell-mediated immunity, which is known as cellular immunity, we will use the T cells. So the humoral immunity will secrete the antibody that defend against the extracellular pathogens, while the cellular immunity will defend against the infected cells itself like cancer and also.
My name is Hanisa binti Muhammad Yaqub and I will present the first homeostasis, which is phagocytosis. So these are the steps on how phagocytosis works. The first one, phagocytes recognize the pathogens by the pathogen-associated molecular patterns (PAMP) displayed on pathogens' cell surface and through receptors on phagocytes' cell surface called pattern recognition receptors (PRRs). Second, phagocytes adheres to the pathogens. Third, phagocyte engulf the pathogens forming a phagosome, phagocytic vesicle. Fourth, lysosome fuses with the phagosome, forming a phagolysosome. Fifth, lysosome secretes lysosomal enzymes to digest the pathogens or debris, forming a residual body containing indigestible material. Sixth, exocytosis of the vesicle remove indigestible material. Chronic granulomatous disease or CGD is an inherited disorder of phagocyte dysfunction caused by defective respiratory or oxidative bursts in neutrophils and monocytes. During the phagocytosis of microorganisms, a respiratory burst occurs, which is characterized by a rapid increase in the formation of reactive oxygen species. Respiratory burst is an essential component of innate immunity, allowing phagocytic cells to kill bacteria. Phagocytic respiratory burst produced by NADPH oxidase 2 in amphioxus. CGD is caused by mutations in any of the 4 genes that comprise the ADPH oxidase complex in phagocytic cells, which disturbs the production of phagocytic respiratory burst. Due to this reason, phagocytes are unable to fight invasive infections because they lack the ability to produce microbicidal oxygen species. The body is vulnerable to life-threatening invasive fungal, bacterial infections, and granuloma formation in vital organs such as skin, lungs, gastrointestinal tract, lymph nodes, liver, and spleen. Nitroblue-tetrazonium or NBT test is used to diagnose CGD. NBT test is a blood test that measures the immune system's ability to change the colorless nitroblue-tetrazonium NBT to a deep blue. The procedures: first, a blood sample is required. Second, in the lab, the chemical NBT is added to the white blood cells. And third, examine the cells under microscope to see if the chemical has made them turn blue. These are the results. If it changed to deep blue color, it is positive NBT because suspension oxygen compounds that kills bacteria are present. This indicates that the respiratory burst is intact. But if it remains unchanged, then it is negative NBT that indicates the people has CGD. This is because of insufficient production of oxidants needed to kill the bacteria due to the dysfunction of NADPH oxidase enzyme complex.
My name is Atika and I will present homeostasis 2, which is inflammation. The inflammatory response or inflammation occurs when tissues are injured by bacteria, trauma, toxins, heat, or any other cause. Inflammation involves in 3 steps. First is inflammatory chemical release. The chemical released by injured or stressed tissue cell and immune cells. The damaged cell release chemicals including histamine, kinase, prostaglandins, and complement. This chemical cause blood vessels to leak fluid into the tissue, causing swelling, and this helps isolate the foreign substance from further contact with body tissue. The second step is vasodilation to increase vascular permeability. It leads to greater blood flow to the area of inflammation, resulting in redness and heat. It causes redness and heat due to hyperemia when local arteries dilate. It also has exudation, which is fluid, proteins, and red blood cells, and white blood cells escape from the intravascular space as a result of increased osmotic pressure and increased hydrostatic pressure. The first diagram shows the exudation that has led to an outpouring of fluid with fibrin into the alveolar space. The second diagram shows the process of exudation aided by endothelial cell contraction and vasodilation, which typically is most pronounced in venous. Chemical mediators producing endothelial contraction include histamine, leukotrienes, bradykinin, platelet activating factor, and the C3a and C5a components from complement activation. Chemical mediators that promote vasodilation include histamine, prostaglandins, and nitric oxide. First, the last step is phagocytosis mobilization. It occurs in four manifests. First is leukocytosis, which is neutrophils are released from the bone marrow in response to leukocytosis-inducing factors released by injured cells. The second phase is margination. Neutrophils cling to the walls of capillaries in the injured area. Next is the diapedesis. Neutrophils squeeze through capillary walls and begin phagocytosis. And the last phase is chemotaxis. Inflammatory chemicals attract neutrophils to the injury site. What the effect if the inflammatory response is disturbed? With the inflammation as a serological response, wounds will fester and infection could become deadly. However, in some diseases, the immune system fights against the body's own cell by mistake, causing a harmful inflammation. For example, inflammatory process goes on for too long, or the inflammatory response occurs in place where it is not needed, so it can become problematic. These include rheumatoid arthritis, where many joints throughout the body are permanently inflamed, psoriasis, a chronic skin disease, inflammatory bowel disease like Crohn's disease or ulcerative colitis.
Assalamu alaikum warahmatullahi wabarakatuh. My name is Anas Adil Muhammad Qasim, and I will continue this presentation with inflammation diseases. Now that we know how important the inflammatory process is in treating wounds, injuries, and so on, we need to know what will happen if the inflammation process gets disrupted. Sometimes external factors may affect the levels of inflammation, which leads to occurrence of diseases such as heart diseases, cancer, and diabetes. And if the situation gets too bad, it may lead to death. Diabetes is an example for inflammation diseases. It's a metabolic disease that causes high blood sugar. Normally, the hormone insulin moves sugar from the blood into cells to be stored or used for energy. However, with diabetes, the body either doesn't make enough insulin or can't effectively use the insulin it does make. If the high blood sugar doesn't get treated as soon as possible, it can damage nerves, eyes, kidneys, and other organs. There are different types of diabetes. We have type 1 diabetes, type 2 diabetes, pre-diabetes and gestational diabetes, and each of them works differently. Type 1 diabetes, also known as insulin-dependent diabetes, is a chronic condition in which the pancreas produces little or no insulin. The exact cause of this condition is unknown, usually because the immune system itself, which normally fights harmful bacteria and viruses, mistakenly destroy the insulin-producing cells in the pancreas. Type 1 diabetes has so many symptoms such as increased thirst, frequent urination, blurred vision, and many others. For diagnosing type 1 diabetes, we have different types of tests. First, we have glycated hemoglobin test. This blood test indicates the average blood sugar over the past 2 or 3 months. Second, we have random blood sugar test. A blood sample will be taken at a random time and can be confirmed by repeated testing. Third, we have fasting blood sugar test. A blood sample will be taken after an overnight fast. Unfortunately, type 1 diabetes has no cure yet. The patient should take insulin and monitor their blood sugar frequently. Also, take care of their bodies and eat healthy food.
Conclusion from the immune system is the immune system will respond to the recognized foreign substance which is known as pathogen. Pathogen that recognized will be eliminated by the innate and adaptive immunity. Innate immunity and adaptive immunity have its own way to eliminate the pathogens. So this picture shows us how the immune system works on our body to protect each and every part of our body. There are some references that we are refer for the immune system. That's all from us. Thank you.